try to get this thing to work. Okay. Before I move on, when you think about the memory, right, you have multiple bits of information. When you thought about the computer memory, for example, when you say 1 byte is 8 bits, right? So now I'm going to tell you the idea and some implementation that we have done in the past to get 1 bit of memory to work inside the living cell. All right, let's think about this. So this is a device that we made. This line, the black line here, represents the DNA, a piece of DNA that we can put inside the cell. The 2 triangles here are the recombination site. So it's a site that some special enzyme that I'm going to tell you in the few slides can bind and do something with it. The piece in the middle here is a promoter. So basically, it's a special sequence on the DNA that tells you where to transcribe. Right? Now, the way that we encode a single bit, the state 0 and state 1, is that we encode it in the orientation of this promoter. So you can go from this state to this state, for example. So this represents state 0 and state 1. Right? Now, let's say if I have any kind of gene downstream, for this example, I have GFP here and RFP here, for example. So if my promoter points this direction, then I can express GFP. Right? On the other hand, if I flip the promoter to the other direction, then I can express the other gene, which is RFP. And I can encode this as a state 1 and the other one as a state 0. Right? Now, the process of flipping this piece of DNA, we use the enzyme called integrase. So this is the enzyme that we borrow from bacteriophage. So if you remember from basic biology class, for some kind of virus, it has an ability to insert a genome into the host genome. And this is basically the enzyme that the virus uses. But we apply it for a different application. So basically, the way it works is that we use this enzyme that we call integrase to flip this bit from state 0 to state 1. Okay? Now, here's how it works in practice. So, when I show you here, the upper one here, it's just a schematic drawing showing the input that we put into the cell. This one is the flow cytometry result. This axis is GFP, this is RP. So each different dots here represent each different cell, telling you, how much GFP each cell expressed and how much RP each cell expressed. All right? And this one is a microscopy image. So basically what I show you here is that the cell at the original state, state 0, and the green cell, the cell that expresses GFP. All right? And you can see here from the cytometry result that you have a lot of GFP and a little RP. Right? Now what happened next is that we induce it with some inducer that allow us to express the integrase. And what happened is that you can see here that all the cells from green state turn red. All right? And you see from the cytometry result that you have all this population move from all green and no red to all red and no green. All right? So at this step, it's just like any kind of inducible promoter that you might have been using in the past. But the nice thing is that once you remove the input, then the state is remembered. Now you remain at state 1 because now the input is gone. You no longer have those integrase expressing in the cell, but the cell still remain red. Right? Here's how you go from state 0 to state 1. Now, if you want to go back from state 1 to state 0, the way it works is that we use the same kind of enzyme, the integrase, but you also need another cofactor. No, it's not really a cofactor, it's a protein that interacts with integrase. It's the protein called excisionase. And when it binds to integrase, it changes the directionality of the reaction. And basically, these two guys working together allow us to flip from state 1 to state 0. All right? And what works here is that you can see here, I start from the red cell. It's pretty difficult to see, but it's red. You can see from the cytometry result. And when we induce it, they flip to green. And then when we remove it, then it remains green. Okay? So this is how we write it from state 1 to state 0. Okay. But these two separate slides that I showed you experimental result, those are the two different devices, the set device and the reset device on two separate populations of cells. So now we need to put them together into the same cell. If you want to get the bit that we can write, rewrite. So if you see this slide, for example, this is the full device. This one is a piece of DNA. This one is also the piece of DNA. This is the bit that I showed you a few slides ago. This is GFP and IFP. And you have two different inducible promoters. If I remember correctly, what we use is a ribinose inducible promoter and tetracycline inducible promoter. But basically, you can use any kind of promoter that you want to induce these two separate capsids. And the way it works, you can see here, this is also the distribution of the fluorescent protein. Here I show only GFP, but not IFP. So what I have here is that here is the different generation time, and here is the level of GFP. All right. The way it works here is that I induce it with the reset signal. So basically express this excisionase and integrase, and basically I flip the promoter from the green state to the red state. Oh, no. From the red state, basically, to the green state. So the level of green is up here, right? And after I remove the inducer signal, it remains in the green state, okay? And I can do the same, induce it, do the opposite thing, basically induce it with the set signal. So basically now, I flip it from the high green state to the low green state here, all right? When I remove the signal and wash it out, now there's no longer input, and it remains in the low green state here. And I can repeat the process over and over in many different multiple cycles, right? And I can also do the same using a little shorter pulse. So this one here is about like the cell has been induced overnight, but this one the cell has been induced for about 4 hours for each induction pulse. You can still see that it can go back and forth over multiple cycles without a change in the performance. So basically, almost every cell switch the state correctly for each cycle. All right. Okay, but this is just one thing, right? But let me tell you why this is important and why we just try to get this paper out. So the storage lifetime is pretty long. For now, we show that at least 400 generations, the state that we write, either 0 or 1, it remains inside the cell. For the base memory that you have been built over the past, have been building over the past, let's say, 20 years, most of them, the state can last for, let's say, 20 to 30 generations. But we show here that our state can last for at least 100 generations. Another thing is that, this is another important thing, we can rewrite the state. So most of the memory that people have built in the past, there are some idea in the past that people use some other kind of enzyme to flip a piece of DNA, but they don't have the capability to rewrite the state. All right. But here we show that we can write and rewrite and write and rewrite at least 8 cycles, and there's no change in the performance. And another thing that it might be pretty subtle to understand is the modularity. So if you look at this schematic diagram, all these things are piece of DNA, right? And basically, everything here, you can swap something else inside of it. So here, you don't have to use GFP. You can use any genes. You can put any genes here. You can use the gene that, for example, control the shape of the cell, or control the fitness of the cell, or control the production of something. You can put any kind of this thing up here and down here, right? And also, up here, you also can put any kind of promoter up here. You don't have to use the ribinose inducible promoter and the tetracycline inducible promoter that I use. You can use any kind of promoter. And the idea that you can put anything here, you can imagine that we could possibly put these bits next to each other, right? You can use the output here to drive the input of the other bits. If you have the next bit, they can operate independently. And so why these two things are important? So let's think about this. Most of the memory that people have been building over the past several decades, it cannot be rewritten. So if you want to store multiple bits of information, the capacity, the number of states that you can store, is only scaled linearly with the number of bits that you have. It counts as thermometer code. So I start from 0, 0, 0, this is like 4 bits in state 0, right? When I go to the next state and put 1 here, and put 1 here and 1 here, right? So if you have 8 bits, it can count up to 8. But if you have capability to write and rewrite and write and rewrite, and also string all those bits together, then you can scale it up to a combinatorial counting. So basically, this guy can count like a binary, like the base 2 number, right? So basically, if you have 8 bits, I can count up to 2 to the 8. We haven't gone that far yet if you can string the bits together, but we already have some plausible composition scheme that we think of to string all these bits together. And we have some preliminary theoretical study on how we can string them together. But right now in the lab, we're on the process of figuring out how to build the next bit and string them together. Because they can count up to multiple states. Okay, so those are my works so far. All right, but since I'm here to represent the synthetic biology community, so I will also talk about other cool stuff that our friends have been making over the past decade. So let's see here. So if you remember the slide that I showed you early on, the idea that you can build something at the different level of abstraction, from the piece of DNA to the parts like a promoter and everything, and you can string them together into a device, right? So the key research area in synthetic biology is basically divided like this. So let me give you some example of the exciting stuff that you have. Let's start from, and also, all these kind of things need to operate inside the living cell, right? So another important research area is to figure out the way to engineer like the chassis, or like, it's like the computer that you can put any kind of program in it, right? How to engineer the cell that you can put this device on. Now, let me go through a few examples in each of these research areas, start from the device. So you see here, this one is a research project from UCSD. So basically, this guy tried to make an oscillator. The first in vivo oscillator that people made was in around, I think, in year 2000. So those ones are like a single oscillator that can operate inside the living cell, a single cell. And you can have the bacteria, like an E. coli, that can blink like a Christmas tree. But this guy, over the past 10 years, they have developed and refined these tools so that it become more reliable. And they also figured out the way that you can have different cells that can communicate with each other, so that all the cells in the population can oscillate at the same time. So the cool thing that I want to show you here, these are the population of the E. coli, and they communicate to one another with some chemical signal, and they oscillate together. This one is, I think it's a GFP signal. So basically the whole population glow, and then turn dark, and then glow again over a pretty regular cycle. And you can check out this paper for like the movie that they put online. It's pretty amazing. Another example is that people try to build a logic gate inside a cell, so that you can integrate different kind of signal. Let's say if I have light and sugar, for example, I want the cell to do this. Or if I have some salt and mechanical input, I want the cell to do that. So an example here is some RNA device. It's the RNA that can sense different kind of small molecule. This example here, I think it's the theophylline, and I'm not sure what's TC here. I think maybe tetracycline. And what they have here is that, for example, this guy is a piece of RNA that can operate inside the cell and function like an AND gate. So the way it works is that if you have only one input, this guy or this guy, there's no output coming out. But if you have both of them, then two to express something downstream. So it's an AND gate because you need two inputs in order to have an output. Or like this guy is a NOR gate, for example. You need to have you need not to have both inputs so that you have some output out, for example. Now, another example here. This one, some of you might have seen it before. So this guy tried to make like a device that allows the cell to communicate to one another and form some pattern. If you think about the cell in your body, for example, when you create like your